Welcome back, everyone. We are right now at the excretory system, the kidneys, and we have the skin at the bottom here. So far, we have done the immune system, the digestive system, the circulatory system, and the respiratory system. Once we finish the excretory system, the kidney and the skin, we have the essential fundamental understandings of the Jack Weston passages on the excretory system and digestive system and the circulatory system with the respiratory and immune system. So we can answer the questions, do some practice questions on the side with these, so that will be helpful. Then we will have the muscular system and the genetics to finish biology. However, we still have, if you remember, the chapters 1 to 5. So yeah, these shouldn't take that long, I believe. So yeah, we can definitely get through them and see how we finish them. But let's now just focus on the excretory system. So right now, as you can see, we have, let's start with the overview of the whole kidney. And yeah, as you can see, we have the excretory, the urine pathway. We have the kidneys, we have the bladder, and we have the nephron. So I just went on and studied these two first sections. So the excretory pathway. So far we have had pathways for the cardiovascular system, the blood pathway. We have had one for the respiratory system and one for the digestive system. Now this is the fourth pathway. There was no pathway for the immune system, as you know. So this is the fourth pathway that we are going to need to know, and I believe there's no more pathways along the road. So this is the fourth and last one. Now for the excretory system, the urine pathway, it starts with the Bowman's capsule, where the filtration happens between the blood vessels, the capillaries, and then we start from the proximal convoluted tubule, and then we have the descending limb of loop of Henle, and then we have the thin ascending limb, and then we have the thick ascending limb, and then we have the distal convoluted tubule, and then we have the collecting duct, and then we have the ureter, and then we have the bladder, and then the urethra. I just missed the renal pelvis. So after the collecting duct, we first go through the renal pelvis. That's where the urine is. Dumped. So the kidneys produce the urine, and they contain a cortex, which is the outer layer, and the medulla, which is the inner layer, and they produce urine, which dumps into the ureter at the renal pelvis, and urine is then collected in the bladder until it is secreted through the urethra. Now, nephron is the functioning unit of the kidney, just like as neurons that are the functional unit of the nervous system. And we have a renal portal system. So we had a portal system for the liver, which is called the hepatic portal system. Now let's see how these two are similar or different or different. We do have the digestive system right here beside us. And we have liver here, as you can see. So what did liver do? Liver synthesizes bile, clotting factors, and one more thing, which was, I believe, the albumin. Yes. And it processes nutrients, so it detoxifies the ammonia in our NH3 made in our body to urea, as well as alcohol and drugs. And liver receives blood from the abdominal portion of digestive tract. So we have an abdominal portion of our digestive tract via the hepatic portal vein. So we have the portal system for our liver as well. So anything that is with hepatic, it's connected. It means that of the liver, and so liver receives blood from the abdominal portion of the digestive tract via hepatic portal vein. Know that liver is receiving blood via the hepatic portal vein. So veins are not usually the ones that give blood to organs, but so you can see here that the portal vein, the hepatic portal vein, is what gives liver's blood. Now let's see if there is something similar for the renal portal vein or renal portal system. So two capillary beds in series, glomeruli and nephron. So the portal system of our kidneys, it has two capillary beds in series, one from the glomeruli and one from the nephron. So nephron, we know it's a functioning unit of the kidney. Now blood flow goes from the renal artery. So the renal artery brings blood. It's not like the hepatic portal vein. It's not a vein, it's artery. And then it has afferent arterioles. We know A as afferent is when it arrives, so it enters the kidney. Then there's the glomeruli. Then there's efferent arterioles. These efferents means that they exit the place. And then we have vasa recta, which surrounds the nephron, and we have renal vein at the end. So the vein is what exits the kidney. Now, this was the renal portal system. Then we have filtration. So there's Bowman's capsule, which moves solutes from blood to filtrate. So at the Bowman's capsule here, we have the solutes moving from the blood, as you can see here, to the filtrate. So this is our filtrate, the whole nephron, basically. The direction and rate is determined by hydrostatic and oncotic pressure differentials between the glomerulus and Bowman's capsule, Bowman's space. So between the glomerulus and the Bowman's space, there is a difference of pressure, both oncotic and hydrostatic pressure. We will come across this. We will definitely get more details about this with our flashcards. So For now, let's just keep in mind that there is a filtration that takes place, and this filtration is the function of the kidney. Then we have secretion, which is the movement of solutes from blood to filtrate anywhere other than Bowman's capsule. So anywhere else other than Bowman's capsule is not filtration anymore, it's secretion. Then we have reabsorption, which is the movement of solids from filtrate to blood. So if you remember ADH and aldosterone, these two hormones do have reabsorption functions. So if you remember, ADH reabsorbed so aldosterone, so which one did change the osmolarity? So aldosterone reabsorbed sodium, which also resulted in bringing in water. However, ADH only reabsorbed water by introducing aquaporin channels in our collecting duct, which resulted in the reabsorption of water only. So the blood osmolarity changed with ADH, but it was reduced. But with aldosterone, it was not reduced. It was not changed because with the sodium, there was also water coming in, right? As you can see here, this is exactly what we were talking about. So aldosterone is a steroid hormone synthesized in the adrenal cortex in response to angiotensin II or high potassium concentration. It is derived from cholesterol and increases sodium reabsorption in the distal convoluted tubule. So yes, sodium is reabsorbed for aldosterone. We got it right, and it happens in the distal convoluted tubule and collecting duct. So it's both of those sites, thereby increasing water reabsorption as well. This will result in increased blood pressure, but no change in blood osmolarity. But ADH, on the other hand, increases blood pressure and decreases blood osmolarity as the urine is concentrated. So it is a peptide hormone. It's not a steroid hormone like aldosterone. And it is synthesized by hypothalamus and the posterior pituitary. It is where ADH is released. It increases permeability of the collecting duct to water, which increases water reabsorption. This results in increased blood pressure and decreased blood osmolarity. So as you can see, because it brings back water into our blood, the blood osmolarity is decreased because we have the ratio of solutes to the solvent is decreased as you have increased the denominator of the ratio, not the numerator. So once you do that, your osmolarity is decreased, and also your urine is concentrated because you took out the water from it. I believe this would happen when you, for example, have a colorful urine, and that is because there's not much water inside it. So we can actually ask ChatGPT later, does concentrated urine mean that it is more colorful than when water is not reabsorbed by ADH? we just drink too much water. Okay. Now, just after secretion, so we know that anywhere else that filtration takes place, so the solids move from the blood to the filtrate anywhere else than the Bowman's space, it's called secretion. Then we have reabsorption, which is the movement of solids from the filtrate to the blood. And we have the pH. The kidney can regulate pH with bicarbonate and H+. So this is probably the third place that we can see how pH could be regulated. It is with the what is the equation called? The bicarbonate buffer? Yeah. Is there another name for the bicarbonate buffer? Does this happen with in blood, lungs, and kidneys? Does the buffer happen in blood, lungs, and kidneys to regulate pH anywhere else than these two? I mean, for blood, it's the heart with it as well. And one interesting question I have in mind is if the pH of the kidneys are any different than anywhere else. So what is the pH at? So what pH are we talking about? What is the pH of blood and filtrate in kidneys? Then let's talk about the nephron. So nephron is this whole thing itself. So it has a proximal convoluted tubule. Oh, one interesting fact about nephrons I remember is that there is 1 billion of them in each kidney. So I hope that number is still intact and has not changed. So we have the proximal convoluted tubule, which is PCT. It is the site of bulk reabsorption of glucose, amino acids, soluble vitamins, salt, and water. Notice that it's soluble vitamins. It means that they are water soluble. So that is B and C vitamins. They are, so PCT is the site of also secretion. So secretion is from the blood to the filtrate, which happens outside of the filtration, that is the Bowman's capsule. So you see, this is an example. PCT is an example of a site outside of, other than the Bowman's space. So it is a site of secretion for what solutes? For H+, K+, NH3, and urea. So you can see here that the solutes go from the blood to the filtrate. So H+, K plus NH3 and urea. Now we should see and make sure that these arrows are in the right direction. So for example, here we have HCO3 minus, bicarbonate, NaCl, sodium chloride, water, nutrients, and K plus, potassium, exiting the nephron, going into the kidney, right? And H plus and NH3 entering the nephron. So another thing is when we're talking about the solutes going from the blood to the filtrate, is this about, so what is the filtrate? Is it the nephron, or is it, so when we are talking about the solutes moving from the blood to the filtrate in our kidneys, is the filtrate the nephron or the cortex or medulla of kidneys, or both? Then the next question, what solutes are entering and exiting from the nephron in PCT here? And then we can ask for the descending limb, the ascending limbs, the DCT or collecting duct. Then we have the descending limb of loop of Henle. So for ascending is also limb of loop of Henle. Now the descending is permeable to water but not salt. Therefore, as the filtrate moves into the more osmotically concentrated renal medulla, water is reabsorbed from the filtrate. So as you can see, as you're going down, because the inner medulla is more concentrated, you filtrate the water out of the you move the water out of the filtrate. So as the filtrate moves into the more osmotically concentrated renal medulla, water is reabsorbed from the filtrate. So the filtrate is going down and the water is reabsorbed. So once it is reabsorbed, it goes back to the kidney and hence our blood. Right? Whatever you are reabsorbing, it goes back to the blood. That's so far that what we know. Then we have the counter current multiplier system. It is the vasorecta and nephron that flow in opposite directions, creating a counter current multiplier system that allows maximal reabsorption of water. So you have maximal reabsorption of water due to this counter current movement multiplier system. Now you have the ascending limb, which is permeable to salt but not water. Therefore, salt is reabsorbed both passively and actively. So salt is reabsorbed both passively and actively, but was water reabsorbed actively? I don't think so. So is water reabsorbed passively or actively in our descending limb of loop of Henle? We know for NaCl, for sodium chloride, it's both passively and actively. The diluting segment is in the outer medulla. So it is in the outer medulla that is it's a diluting segment. Because salt is actively reabsorbed in this site, the filtrate becomes hypotonic compared to the blood. So because salt is actively reabsorbed in this site, the filtrate, the nephron going up, is actively, the filtrate becomes hypotonic. Hypo means below, it's less compared to the blood. So tonic comes from probably the concentration of the salt. So when it's hypotonic, because the salt is going to the outer medulla, it is being diluted, right? So once it's going up, it's being watery again, right? You are diluting it. So the filtrate is diluted because salt is actively reabsorbed in this site into the outer medulla, and the filtrate becomes hypotonic compared to the blood. So it becomes hypotonic. If salt would have been reabsorbed, reabsorbed back into the filtrate, if it had been not moved from the filtrate into the outer medulla, then it would have been hypertonic compared to the blood. But what happens here in the ascending limb is that you have NaCl, the salt, being reabsorbed back into outer medulla, so the filtrate is diluted, and as a result your filtrate is hypotonic compared to the blood. And then you have DCT. It is responsive to aldosterone and is a site of salt reabsorption and waste product excretion like the PCT. So you have salt reabsorption. We know now that sodium chloride, salt, is reabsorbed in both PCT and DCT and the thick ascending limb loop of Henle. Therefore, these sections, the medulla is much more concentrated than the cortex. However, still it is a site of the cortex is still the site of PCT and DCT. So salt is reabsorbed back into our kidneys, and the waste product is excreted like the PCT. So we have waste product excretion. So excretion is when we excrete urea and H2O, water. Now, the collecting duct is responsive to both aldosterone and ADH and has variable permeability, which allows reabsorption of the right amount of water depending on the body's needs. So the collecting duct is responsive to both aldosterone and ADH. Yes, perfect, we know that, but the DCT is only responsive to aldosterone. Now, it has variable permeability, the collecting duct, which allows reabsorption of the right amount of water depending on the body's needs. So whatever amount of water your body needs, it will reabsorb it through the collecting duct. It also still reabsorbs urea and sodium chloride. But where does this go after? So we are saying that here, after the collecting duct, the urine, so this is all for the urine. The urine goes to the renal pelvis, and then ureter, and then bladder, and then urethra. So basically, all this filtration that's happening, this filtrate is going to be our urine at the end. So keep in mind that when you are reabsorbing salt, you're reabsorbing it back to the body, not the urine. So the whole point of excretion is that you want to get rid of the components, so you have urine that is being excreted, right? Now, why should you excrete, why should you reabsorb urea, right? So you saw here that, for example, with DCT, you have waste product excretion. So waste products are being excreted via the urine, right? But the salt is being reabsorbed, right? Now, that's also what aldosterone does. So you want to be able to regulate your blood pressure and also be able to regulate the blood osmolarity. You don't want too much. You don't want to dilute your blood too much, or make it too concentrated, right? You want to have it all under control. That's why you reabsorb salt to your need. That's why you excrete waste products. And just we need to know if so this urea should be related to the not Krebs cycle, but the urea cycle. So let's quickly look at the urea cycle. So we have the urea cycle here, and then citric acid cycle, and here is the urea cycle. So this occurs in the cytosol and mitochondrial matrix of the liver, not the kidney, right? So you have here four main substrates as arginine, argininosuccinate, citrulline, and ornithine. And what happens is that water is turned into urea, and you have the fumarate that goes into the citric acid cycle, and you have the citric acid cycle that gives the oxaloacetate and aspartate for the urea cycle, and then you have also carbamoyl, phosphate, and CO2 and NH4+, which is ammonium, into the urea cycle. So these are all to keep in mind while we are talking about the excretion system and urea and urine. So also we don't want to confuse urine with urea. So let's ask a question. What are the components excreted via urine? What is urea, and why is it reabsorbed by our collecting duct, or even PCT? yeah, we need to know also is it reabsorbed by PCT? Because we're not sure here. We don't have urea here. So for now, we did cover the excretory system. Now we need to go for so we do also have the skin, so let's go quickly over the skin. It's a relatively very low yield concept topic, but still you need to know it, right? So we have the epidermis, dermis, and hypodermis, which is the subcutaneous layer. Now, epidermis has 5 subsections. There's also dermis that has all these 6 sections. Then there is hypodermis, which is fat and connective tissue, which connects the skin to the body. Now, epidermis has 5 different stratums: stratum basale, stratum spinosum, stratum granulosum, luciderm, and corneum. BSGLC. Now, basale is where stem cells are turned into keratinocytes. Then spinosum is where the Langerhans cells, I hope it's Langerhans, not Langerhans, cells lie. There was something about Langerhans cells that I think we needed to know in one of the, like, probably in the immune system. But we will see. Then I hope it's not related to defensins. I mean, I actually might hope so. It makes my job easier. But yeah, we have granulosum, which is granule comes from like pigments. So keratinocytes die. That's where we get the pigments, also the color of our skin from, probably. Then we have luciderm, which is only on thick, hairless skin, like the sole of our hands or foot, I would imagine. Then we have the corneum, which is multi thin layers, flat keratinocytes. Then we have the Langerhans cells. See, these are macrophages that are antigen presenting cells in skin. So they present antigens in skin. It should be still part of the innate immune system, not the adaptive. So let's see so let's add skin here. Are Langerhans cells part of the innate or adaptive immune system? Then we have so what were defensins again? As far as I know, they were antimicrobial. However, on the skin and like mucus, I believe. Let's take a quick look here. We had these are good also for our revisions. So we had defensins. So our skin secretes antimicrobial enzymes like defensins. So yeah, these are antimicrobial enzymes. Now, if needed, we'll come back to it, but what cells secrete defensins? Then we have melanin, which is produced by melanocytes. They protect the skin from DNA damage caused by ultraviolet radiation. Then we have dermis. So melanin is produced by melanocytes, and they protect skin from DNA damage caused by UV radiation. So you have UV radiation that might cause damage to the DNAs, and however, we have melanocytes that protect the skin from that, and melanocytes produce melanin. So melanin is what protects skin from DNA damage. Now, how does melanin protect the skin from DNA damage by UV radiation? But then we have dermis, which is the papillary layer, and it has a reticular layer. So it's also written here sensory. So we have Merkel cells. So these are for the cells for our sensory perception. We have Merkel cells, deep pressure and texture for that. We have free nerve endings for pain. We have the Meissner cell corpuscles, which is for light touch. We have Ruffini endings for stretch, and the Pacinian corpuscles for deep pressure and vibration. So Merkel is for deep pressure and texture, but Pacinian is for deep pressure and vibration. We need mnemonics for these. So need mnemonics for epidermis and dermis. We have the hypodermis that connects the skin to the body, and it consists of fat and connective tissue. Now we have thermoregulation, which is happening by 4 mechanisms. We have sweating by evaporative cooling. We have piloerection for warming. We have shivering for warming. We have vasodilation, vasoconstriction for cooling and warming. So when you want to cool down, you will have vasodilation. So you want more blood to release heat. But when you want to keep the heat, keep more blood in, you do vasoconstriction, right? Now, when you want to warm yourself up, you shiver. When you want to also warm yourself up, you have piloerection. So piloerection means that your hair is becoming straight. You have goosebumps. How does it make sense that it's warming you up? How does piloerection warm one up? Okay, let's get to the flashcards. We have 1A custom study. Let's see how many So we have 96 cards. 96. Put 97. New cards. Choose tag. We have this one. We don't want the other ones. It's the biology. What are these? These are like okay, the tags, you know, like Pixarized Khan Academy. Yeah, like I believe what we select from the Kaplan has all of that. So and it's B10, I believe. Kidney and skin. So this is an example of what epithelial tissue. This should be, because it's squamous, I believe, simple squamous. This is an example of squared, simple cuboidal. This is an example of like rectangular, simple columnar, cuboidal, squamous, columnar. This is an example of pseudo-stratified columnar epithelial tissue. This is like stratified squamous. So this is stratified squamous. The one liner was the simple squamous. This is stratified cuboidal because it's 2 layers, stratified cuboidal. And this is an example of stratified columnar. Yes. This is transitional. So once you have all these squamousy, cuboidal, columnar all together, is transitional. Bone, cartilage, tendon, and blood are examples of what tissue? They are connective tissue. It's bone also included. So we have dense connective tissue. We have compact bone connective tissue. We have the blood as connective tissue. We have adipose tissue. We have areolar tissue, the connective tissue. So we have bone, cartilage, tendon, and blood as examples of connective tissue. We got this right. So epithelial tissue covers what and what surfaces of the body? It should be covering internal and external surfaces of the body. So it covers both internal and external surfaces of the body. So the excretory urine pathway goes from the Bowman's space, Bowman's capsule, the PCT, descending limb of loop of Henle, the ascending, the thin ascending, the thick ascending. Then we have the distal convoluted tubule. Then we have the collecting duct. Then we have, the ascending loop is same, so we have the collecting duct, and then we have the renal pelvis. We have the ureter, and then we have the bladder, and then urethra. urethra, bladder, ureter, renal pelvis, collecting duct, DCT, ascending limb, descending limb, PCT, involvements, capsule. We have minor calyx here. We have the nephron. We have the collecting duct that ends here, so it goes to the minor calyx. We have the renal pelvis that comes here. We have the major calyx, and this is the renal pelvis. And then we have renal artery, vein. It is just the same as any other artery and vein. The artery brings oxygenated blood, and the renal vein takes out deoxygenated blood. We have the ureter that is here, then urinary bladder, the frontal section of right kidney. Amazing. We have also this diagram, the healthy kidney versus the diabetic kidney. So the principal cell is here, intercalated cell is here, there's the urine flow, then we have the AMP concentration, and then we have the effective microbial barrier, we have UPEC infiltration, we have the insulin dependent AMPs, but we don't have them in a diabetic kidney. Then we have AMP targeted UPEC. You have insulin independent AMPs, and yeah, we don't have these insulin dependent AMPs. Then we have UPEC and urethra. Adherent UPECs provoke inflammatory response. Man, I don't want to even look at this. But this YouTube link, I think it's a good one. I remember Bozeman science.
Hi, it's Paul Anderson, and in this video I'm going to go through the basics of the urinary system. The major organs in the urinary system are going to be the kidneys, and their major role is in waste excretion. They're cleaning our blood, and solid waste from the blood is eventually going to leave our body as urine. Now, in addition to doing that, they also regulate blood volume, blood pressure, blood pH. They regulate important electrolytes and metabolites in our body. The kidneys are incredibly important. Without them, you'll die unless you're hooked up to a kidney dialysis machine. But that's going to require going 3 times a week, 4 to 5 hours a day, hooked up to a machine just to clean your blood. Now, in addition to the kidneys, we also have other parts of the urinary system, including the ureter, the bladder, and the urethra. In males and females, the urinary system is essentially identical, except for the length of the urethra itself. Now, what's the role of the kidney? The role is to produce urine. From there, it's transported to the bladder through the ureters, and then from there, it's stored and eventually conducted into the environment. That's how we lose this waste. Now, you might be thinking to yourself, what is urine? Well, urine is mostly water, so 90 to 95% of urine is going to be water, but the rest of it is going to be solid waste, both inorganic and organic. Organic waste like urea, uric acid, and inorganics like toxins and certain salts as well. If we look at the structure,
so the urine waste is 5 to 10% solids and 90 to 95% water. The solids has organic and inorganic component.
Organics like toxins and certain salts as well. If we look at the structure of the kidney, on the outside we're going to have the renal cortex. Renal simply means kidney. So we've got the outer layer of the kidney, and then on the inside we have the renal medulla. And then on the center we have the renal pelvis. This is going to be a hollow opening, and this is where all the nephrons are going to enter into. They're going to get rid of their urine, and that's eventually going to go through the ureter. Now, if we look inside the kidney itself, we find the whole thing is just filled with blood vessels. Around 20% of cardiac output is going through the kidneys. So that's a massive amount of blood that's going through the kidneys. How does it do that? Well, it comes in through the renal artery, out through the renal vein, but there's a huge amount of surface area inside the kidneys itself. From there, we go into smaller arteries and smaller arteries, and then arterioles and capillaries, and then eventually the blood is going to come out. But the important waste excretion is going to take place as we get to those center blood vessels. So let's zoom into that, and let's zoom into the functional unit of the kidney, which is going to be the nephron. So if we look at where these blood vessels are found, blood's going to flow into the nephron through the afferent arteriole, and then it goes into the glomerulus, or the glomerular capillaries. At this point, you essentially have a dead end. The blood is flowing in, and it comes to a dead end. That dead end is inside something called the Bowman's capsule. At this point, we're going to have filtration occurring. A lot of that blood plasma is squirting out of the capillary, and it's eventually going to move into the urinary system. From there, the blood goes through the afferent arteriole. And so at this point, we've got filtration taking place. This is where we're actually filtering the blood. Now, if you look at the blood vessels, they continue to go and wrap around the rest of the nephron, the peritubular capillaries, before they're eventually going to leave. What's going on all of this surface area is secretion and reabsorption. Secretion is when we're taking material that's in the blood that wasn't filtered out, but we're getting rid of that. And also there's some material in the filtrate, glucose, water, for example, that we want to reabsorb. So we're going to take that back into the circulatory system. So if we remove the circulatory system, now we can kind of see what that nephron looks like. First part's going to be the renal corpuscle right here. If we look at what's taking place there, that's where that filtration occurs. So at this point, we call this a filtrate. From there, it's going into a number of different renal tubules. The first one is going to be the proximal convoluted tubule. Proximal means close to the renal corpuscle. Convoluted means it's just folded over and over in many different dimensions. We're increasing the surface area there. What's going on at this place is that we're reabsorbing important material. We're reabsorbing solutes, water. We're taking that back into our circulatory system. From there, the renal tubules are going to flow down into what's called the loop of Henle. We have a descending limb of the loop of Henle, and during that, we're reabsorbing a lot of the water. And then as we have the ascending limb of the loop of Henle, we're reabsorbing a lot of that sodium and chloride. A lot of that salts are coming back into our body. If we look at where we are inside the kidney itself, as we go down into the loop of Henle, we've entered into that renal medulla. And if you think about what's going on as we move farther and farther down, we've set up a countercurrent exchange. What we're doing is we're increasing the salt levels as we move farther and farther down into that renal medulla. That makes it easier for us to take back more of that water in the descending limb of the loop of Henle and also in the collecting duct. From there, we're moving into the distal convoluted tubule and then into the collecting duct. If you think about where this collecting duct is going, you can see that we're getting a bunch of other nephrons that are connected to it as well. From here, it's eventually going into that center of the kidney, the renal pelvis, and eventually it's going to leave as urine. Now, what's going along in that distal convoluted tubule and the collecting duct is we have variable secretion and reabsorption of solutes and water. So in other words, sometimes we're getting rid of waste and sometimes we're taking that back into the body. Now, a lot of this is under hormone control. So for example, a really important hormone is vasopressin or ADH, antidiuretic hormone. And what that's going to do is it's going to affect the distal convoluted tubule and the collecting duct itself. And what it does is it essentially shuts down the flow of water outside our body. And so we can get a signal. If we sense how much water there is in our blood, we can get a signal that means we have to hold on to that. Or we can have a signal that says we want to release that water and it's eventually going to leave as urine. If we look at where it goes, from here, all of that waste that comes out of the nephron is emptying out eventually into the pelvis, into the ureter, and it's stored in the bladder itself. From there, we can eventually conduct that to the environment. And so that's how the urinary system works. It's really simple. It's also really, really important, and I hope that was helpful.
Okay, cool. So we just still need to understand what is the difference between a healthy versus a diabetic kidney. What are AMPs and UPECs involved in the process? Okay. Do we answer this? Yes. So we're good. Identify the missing. I love these missing pieces. PCT in ascending limb. Ascending limb is the descending limb. Very good. The distal DCT in renal pelvis. It's the collecting duct. The kidneys are tubing-shaped organs that help the body pass what as urine and also help, so they help the body pass waste as urine and also help filter blood before sending it back to the heart. So yeah, they pass. And also, as the Bozeman Science channel video said, Mr. Anderson, he said that 20% to 30% of blood comes to our kidneys. So that's a massive amount of blood that needs to be filtered. So it also helps, the kidneys also help filtering blood before sending it back to the heart. And they pass waste as urine. So the dirty blood goes in and clean blood goes out. Are we saying that it's deoxygenated blood that comes in and oxygenated goes out? We need to check that to make sure. Step 1, glomerular filtration. Water, ions, amino acids, glucose, nitrogen containing wastes, and other small molecules move from the glomerulus to the inside of the glomerular capsule to form glomerular filtrate. Step 2, tubular reabsorption. Water, essential ions, and nutrients are reabsorbed from the proximal convoluted tubule into the surrounding capillaries. Some reabsorption of water and ions occur along other sections of the renal tubule and collecting duct. And then step 3, which is the tubular secretion, as wastes, excess ions, and drugs are actively secreted into the distal and proximal convoluted tubules from the surrounding capillaries. Some secretion also occurs along the collecting duct. So super filters that clean your blood, waste leaves the body. Does deoxygenated blood enter kidneys and oxygenated blood exit kidneys via renal vein, via renal artery? Okay, if that's the case, then it is both kidneys and our lungs that bring in deoxygenated blood, and which is artery, and the vein is the one that has oxygenated blood. But I don't think so, since oxygen is not in the kidneys because it's only in the lungs that we have that. But anyway, we will figure that out. What is the functional unit of the kidney? It's nephron. Amazing. There are 1.3 million nephrons per kidney. So my bad. I thought it was in the 1 billion, but it's 1.3 million. To make sure, how many nephrons are there per kidney? Approximately. Why am I writing these under the skin? Okay. The kidney has two capillary beds in parallel or series. It's in parallel. They connect the arteries to the veins inside the nephron. It's in series. So two capillary beds in series that they connect the arteries to the veins inside the nephron. The two capillary beds are the glomerular and peritubular capillaries. Note there are two other instances in the human body of capillary beds in series, the hepatic and the hypophyseal portal venous system. The kidney is unique because there is an arteriole between these, but is still one of three portal systems in the body. So we have the interlobular artery that has the afferent arteriole, and then the efferent arteriole, and then we have the proximal convoluted tubule here. But here we have the glomerular capsule, we have the venule, we have the interlobular vein, the loop of nephron, the peritubular capillary network, the peritubular capillaries, and the glomerular. So the kidney has two capillary beds. These two capillary beds are in series, not in parallel. They connect the arteries to the veins inside the nephron. So both these peritubular and glomerular capillaries then go into the venule and the interlobular vein at the end. What is a network of capillaries located at the beginning of each nephron in the kidney? It's the glomerulus. It's a network of capillaries located at the beginning of each nephron in the kidney. The renal corpuscle. We have the distal convoluted tubule. We have the macula densa. We have the juxtaglomerular cells, afferent arteriole, mesangium, glomerulus capillaries, podocyte, proximal tubule, glomerular capsule, glomerular space, capillaries, red mesh strainer, Bowman's capsule, bowel, glomerulus gloves.
Hi, in this video we're going to take a look at glomerular histology. Histology is the study of the microscopic anatomy of cells and tissues. So we are going to be looking at the glomerulus really close up. I'll start by drawing a glomerulus. So the blood flows through the glomerulus via these arterioles, entering through here and exiting through here. The blood is filtered in the capillaries inside the glomerulus, and the filtrate flows out here. The glomerulus is surrounded by the Bowman's capsule, which is made of epithelial cells, which we call parietal epithelium. Epithelium is one of the 4 basic types of tissue, the others being muscular, nervous, and connective tissue. And epithelium lines the surfaces of structures of the body. Now, between the parietal epithelium and the capillaries, we have Bowman's space. This is the area into which filtrate collects before flowing into the proximal tubule. As we discussed in the last tutorial, these are the afferent and efferent arterioles, and they can constrict and dilate in order to regulate the rate of filtration, known as the glomerular filtration rate, or GFR. And like all blood vessels, these arterioles are lined by endothelial cells, not to be confused with epithelial cells. They are a completely different type of cell and have a different embryological origin. Now, this area here is the beginning of the proximal tubule, and it has these characteristic brush border epithelial cells, presumably to increase the surface area of the cells. And this allows for greater reabsorption and secretion of solutes and water. Between the 2 arterioles is this distal convoluted tubule, which is rising up towards us at right angles with the plane of the page. And here, covering the capillaries, we have a cell called the podocyte. It's called a podocyte because it has feet, and pod means foot in Greek. And these are also a type of epithelium. We call these visceral epithelium. We'll talk more about them later in this tutorial. Between the distal convoluted tubule and the glomerulus, there is a highly specialized group of cells called the juxtaglomerular apparatus. Again, not to be confused with the juxtamedullary nephron, which is something else entirely. So I want to talk about the juxtaglomerular apparatus because it is super important. There are 3 cell types in the juxtaglomerular apparatus. The first are granular cells, and they produce renin, which is the start of the renin-angiotensin-aldosterone system, which is really important in controlling blood pressure. For more information about this, see the handwritten tutorial on the renin-angiotensin-aldosterone system. The second type of cell is a macular denser cell. This is a cell that detects how much sodium chloride is passing through the distal convoluted tubule and sends a signal to the granular cells to release renin. There are also these extraglomerular mesangial cells, but the function of these is contentious. Now I want to talk about the capillaries within the glomerulus. I'm going to take a cross-section through here and draw what we might find down here. So there are 3 types of cell involved here. The first is the capillary, which again is an endothelial cell. And these are attached to mesangial cells, which are like the scaffolding for the capillaries in the glomerulus. They can actually contract and relax to alter the glomerular filtration rate. The third type of cell is this podocyte, which we saw before, covering the whole capillary network. These prevent large molecules such as albumin from being filtered by the kidneys. And they too can contract and relax to increase the gaps between them and increase and decrease the glomerular filtration rate. And that's an overview of the histology of the glomerulus. For more free tutorials and the PDF of this tutorial,
That was actually useful. Wow, this comment is pretty harsh. Okay, nice. So far, so good. So it is the glomerulus, that is a network of capillaries located at the beginning of each nephron. This is a simple cuboidal epithelial tissue. So we now know that knowing epithelial is completely different than endothelial that is in our blood capillaries. Epithelial is one of the four main tissue types, the other ones being muscular, connective, and nervous, I think.
which surfaces of or GFR embryos and have a different embryological epithelial cells. They are a completely different type of cell and have a different embryological origin. Now this area here is the cells, and they can constrict and dilate in order to regulate the rate of filtration, known as the glomerular filtration rate, or GFR. And like all blood vessels, these arterioles are lined by endothelial cells, not to be confused with epithelial cells. They are a completely different type of cell and have a different embryological. Histology is the study of the microscopus really close to the glomerulus via these arterioles. The blood is filtered in the capillaries inside the glomerulus, and the filtrate flows out here, surrounded by the Bowman, which we call is one of the four basic types of tissue. Epithelium is one of the four basic types of tissue.
Here you go.
The others being muscular, nervous, and connective tissue.
Yeah,
And
so
epithelium lines the surfaces.
yeah, so it's muscular, nervous, epithelium, and connective tissue. So muscle cells, connective tissue, which is blood, bone, cartilage, tendon, and so connective tissue, muscle, nervous system, right? Now this is simple cuboidal epithelial tissue. This is an example of simple squamous epithelial tissue. This is an example of stratified cuboidal tissue. This is a stratified squamous. This is transitional epithelial tissue. This is pseudocolumnar, pseudostratified columnar. This is simple columnar. Epithelial tissue covers what surfaces of the body is internal and external. Bone, cartilage, tendon, and blood are examples of connective tissue. The kidney regulates pH by adjusting the levels of excreted waste and reabsorbed water bicarbonate. It's excreted H plus and reabsorbed bicarbonate. So sodium hydrogen antiport secretes H plus hydrogen. So secreting means from the blood to the filtrate. Secreted H plus from the proximal tubule to the Bowman's capsule. So it's from the blood to the filtrate. H plus infiltrate combines with filtered bicarbonate to form CO2 and water. CO2 diffuses into cell and combines with water to form H plus and bicarbonate. H plus, where's step 4? H plus is secreted again and excreted. Secreted H plus and the NH4 plus will be excreted. Then we have bicarbonate that is reabsorbed here. We have the peritubular capillary, so reabsorb back into blood, into our system. We have glutamine, which is metabolized to ammonium ion and bicarbonate. We have glutamine here. Then we have ammonia, which is secreted and excreted. We have HCO3 minus bicarbonate, which is reabsorbed. This here is where we have the sodium hydrogen antiport. So sodium comes in and hydrogen goes out back into the filtrate to be excreted. So pH is regulated by kidney via the excretion of H plus and the reabsorption of bicarbonate, as you can see here.
pH is an indicator of acidity. The body's blood pH is strictly regulated within a narrow range between 7.35 and 7.45. This is because even a minor change in acidity may have devastating effects on protein stability and biochemical processes. Normal cellular metabolism constantly produces and excretes carbon dioxide into the blood. Carbon dioxide combines with water to make carbonic acid, which dissociates into hydrogen ions and bicarbonate. This is an equilibrium, meaning all of the components of the left and right sides coexist at all times, and the concentration of any component is determined by that of others at any given moment. The rule of thumb is an increase in concentration of any component on one side will shift the equation to the other side, leading to increased concentrations of all components on that side, and vice versa. This equilibrium is central to understand acid-base regulation. Continued carbon dioxide production by all cells of the body drives the equilibrium to the right to generate more hydrogen ions. Because pH is basically a function of hydrogen ion concentration, more hydrogen means higher acidity and lower pH. Normal metabolism therefore constantly makes the blood more acidic. The body must react to keep the blood pH within the normal limits. This is achieved by two mechanisms. Elimination of carbon dioxide through exhalation. The amount of carbon dioxide exhaled by the lungs is regulated in response to changes in acidity. A decrease in pH is sensed by central or arterial chemoreceptors and leads to deeper, faster breathing. More carbon dioxide is exhaled, less hydrogen is made, blood acidity decreases, and blood pH returns to normal. Pulmonary regulation is fast, usually effective within minutes to hours. Excretion of hydrogen ions and reabsorption of bicarbonate through the kidneys. The kidneys control blood pH by adjusting the amount of excreted acids and reabsorbed bicarbonate. Renal regulation is slower. It usually takes days to respond to pH disturbances. Although all of the plasma bicarbonate is filtered in the glomerulus during the first step of urine formation, virtually all of it is reabsorbed back into the blood. Most of this reabsorption happens in the proximal tubule. The amount of reabsorbed bicarbonate in the proximal tubule is regulated via a number of mechanisms in response to changes in blood pH. It increases during acid loads and decreases during alkali loads. While the proximal tubule basically returns filtered bicarbonate back to the blood, the downstream collecting duct generates new bicarbonate by actively secreting acids. As protons are depleted from the distal tubular cells, the equation shifts to the right, producing more bicarbonate, which then exits into the blood. Hydrogen ions secreted into the lumen combine with urinary buffers, mainly filtered phosphate and ammonia, to be excreted in urine. The ammonia buffering system is particularly important because, unlike phosphate, which is filtered in fixed amounts from the plasma and can be depleted during high acid loads, ammonia production is regulated in response to changes in acidity, and its concentration may increase several-fold when necessary. Blood pH is the main regulator of acid excretion, but potassium, chloride concentrations, and several hormones also play important roles. Pathologic changes may cause acid-base disturbances. Acidosis refers to a process that causes increased acidity, while alkalosis refers to one that causes increased alkalinity. It's not uncommon for a patient to have several processes going on at once, some of them in opposite directions. The resulting plasma pH may be normal, too acidic, called acidemia, or too basic, called alkalemia. Acidosis may result from inadequate function of the lungs, which causes arterial carbon dioxide to accumulate. This is respiratory acidosis. On the other hand, metabolic acidosis may result from excessive production of metabolic acids, ingestion of acids, decreased ability of the kidneys to excrete acids, or loss of alkali. Metabolic acidosis is characterized by primary decrease in plasma bicarbonate. Alkalosis can also be either respiratory or metabolic. Respiratory alkalosis is caused by increased ventilation, resulting in excessive exhalation of carbon dioxide. Metabolic alkalosis can result from excess loss of acids through the kidneys or gastrointestinal tract, bicarbonate retention, or ingestion of alkali. Metabolic alkalosis is characterized by primary increase in plasma bicarbonate. Thank you for watching.
Okay, cool. So we got to know more about, but this is a really good 8th step with regards to the kidneys and how they regulate the pH. So, yeah. Aldosterone is a hormone synthesized in the, I believe, adrenal cortex in response to low blood pressure or high K plus, in response to angiotensin 2 or high potassium concentration. So we have a renin angiotensin system. If there is a drop in blood pressure or fluid volume, then we have renin release from kidneys. We just got to know that renin is produced by the juxtaglomerular cells, then, or maybe the podocytes. Yeah, like we need to double check that. I mean, it's not needed for MCAT, I believe, but I mean, it doesn't hurt. But renin is released from kidney, then it helps in turning angiotensin into angiotensin 1. Renin acts on angiotensin to form angiotensin 1, then angiotensinogen into angiotensin. So angiotensinogen is a zymogen, which is an inactive form of the peptide. And then we have ACE, which is angiotensin converting enzyme, which is released from the lungs. So ACE, angiotensin converting enzyme, turns angiotensin 1 to angiotensin 2, and then ACE acts on angiotensin 1 to form angiotensin 2, so we get that here. And once we have angiotensin 2 acts directly on blood vessels, stimulating vasoconstriction, narrowing. So this results in increasing blood pressure, right? When you constrict the vessels, you narrow them out, there's more pressure on the vessel. Angiotensin 2 acts on the adrenal gland to stimulate release of aldosterone. Aldosterone acts on the kidneys to stimulate reabsorption of salt and water. So there you have it. Hormones acting on kidney. We also have atrial natriuretic peptide, which is the reverse of ADH and aldosterone. It is secreted in response to increased atrial pressure and causes increased GFR, glomerular filtration rate, and increased sodium filtration with no compensatory sodium reabsorption in distal nephron. The net effect is sodium loss and volume loss, so you decrease the blood pressure. But aldosterone is secreted in response to lower blood volume via angiotensin 2 and increased plasma potassium concentration, K plus concentration, causes sodium reabsorption to be increased in the we know DCT and collecting duct, and the K plus secretion and H plus secretion. Now, ADH vasopressin is secreted in response to increased plasma osmolarity and lower blood volume. It binds to receptors on principal cells, causing increased number of aquaporins and increased water reabsorption. We have angiotensin 2. This is a comprehensive, this pictures the whole interaction between these hormones. We have angiotensin 2, which is synthesized in response to lower blood pressure, causes efferent arterial constriction, which increases GFR and FF. I don't know what's FF, filtration something, maybe. But with compensatory Na plus reabsorption in proximal and distal nephron. Net effect is preservation of renal function in low volume state with simultaneous sodium reabsorption both proximal and distal to maintain circulating volume. Then we have the parathyroid hormone, which is PTH. It is secreted in response to lower plasma, so Ca2 plus calcium, and increased plasma, PO4 3 minus, I think it gets phosphate, or lowered plasma 1,25-OH2 D3 causes increased calcium reabsorption from DCT, decreased phosphate reabsorption from PCT, and increased 1,25-OH2 D3 production. That's 25 vitamin D, and increased calcium and phosphate absorption from gut via vitamin D. So as you can see, the ANP has effect on the afferent DC and DCT, afferent arterial, and then we have PTH, which has effect on the PCT and DCT. Then we have angiotensin, which has effect on the efferent arterial, the PCT and DCT, and aldosterone, it has effect on both DCT and collecting duct, and ADH is only on the collecting duct. stimulates release, inhibits release. We have hypothalamus, the anterior pituitary, posterior angiotensin II, hyperkalemia. You have the adrenal cortex. We have aldosterone, cortisol, androgens. We have mineralocorticoids like aldosterone. We have the glomerular zone. We have the fascicular zone, the glucocorticoids. We have reticular zone, which is where androgens are produced. Then we have the adrenal medulla, where epinephrine and norepinephrine are produced. Then adrenaline and noradrenaline are going to the epinephrine stimulates ACTH release. The adrenocortical T hormone, adrenocorticoid hormone, ACTH, and ACTH is released from the anterior pituitary, which again comes back into circle, affecting on these sections. Stress, basically, sympathetic nervous system, causes both the hypothalamus, CRH, to release that, and also on norepinephrine and epinephrine to have effect on that. Now we have the cortisol and transcordon that inhibit the release of ACTH and CRH, cortico-releasing hormone, and they also blood flow path within the adrenal gland, so they reinforce epinephrine, norepinephrine. I believe it is the transcordon that goes to stimulate the release of epinephrine and norepinephrine. This is a really good diagram in my opinion. This one also is from MCATreview.org. The waste secreted by PCT is NH4 plus ammonia, creatinine, organic acids. Okay, nice. So aldosterone is a hormone synthesized in the adrenal cortex and responds to angiotensin II or high K plus concentration. The kidney has two capillary beds in series. These are the glomerular and peritubular capillaries. There are two other instances in the human body of capillary beds in series. The two other instances are hepatic and the hypophyseal portal venous systems. So we have it in our hepatic, in our liver, and hypophyseal. So this should come from hypophyseal gland, portal venous system. The kidney is unique because there is an arteriole between these. So there is an arteriole, afferent arteriole between these two capillary beds, but it is still one of three portal systems in the body. So we have hepatic portal system and the hypophyseal portal system. So how are the hepatic and hypophyseal portal, hepatic, renal, and hypophyseal portal systems similar? different than each other. Okay, let's move forward. Aldosterone increases sodium reabsorption in the DCT and collecting duct, thereby increasing water reabsorption and raising blood pressure. DCT and collecting duct. Yes, yes, yes. ADH is a hormone that increases permeability of the collecting duct to water, which increases water reabsorption, results in higher blood pressure and lower blood osmolarity. The urine will be concentrated, aka higher urine osmolarity. So the osmolarity for urine is increased, but for the blood is decreased. In contrast, aldosterone reabsorbs sodium, and since water follows salt, so the blood osmolarity does not change as the sodium gets the water to follow it to blood. Perfect, we know that already. So this is where ADH is present. Collecting duct is highly permeable to water, so water is reabsorbed back into our system, into our body, but this reduces the osmolarity of our blood, but increases the osmolarity of urine because urine is concentrated, so small volume of concentrated urine. But when ADH is not present, we have a large volume of dilute urine. So kind of when I am drinking too much water and my urine is transparent, it means that my blood pressure is kind of controlled and is turned lower. Okay, we got that right as well. Urine passes from the kidneys through the what? Then into the bladder, so it's ureter, bladder, and then out through the urethra. Amazing. We have two ureters we have the uterus in women and bladder here and prostate here in men, then urethra. What is a network of capillaries located at the beginning of each nephron in the kidney? It should be the glomerulus. It is a network of capillaries. Yes. What is the muscular lining of bladder? It relaxes to store urine and contracts during urination. It's the detrusor muscle. It is under parasympathetic control because when you are stressed, when you are relaxed, you want to store your, so when you are contracting, you're urinating, so when you're relaxed and under parasympathetic control, do you want to pee more or less? Piss more or less? The internal or external urethral sphincter is made of smooth muscle and is under parasympathetic control. So it should be the internal that is under smooth muscle and under parasympathetic control, because the internal one should be under autonomic control, smooth muscle. And also parasympathetic control. Which one is skeletal muscle is under voluntary control. It is the external. So external is here and internal is there. So this is what we are able to control for let's say is guys. What part of nephron is the site of bulk reabsorption of glucose, amino acids, soluble vitamins, salt, and water? I should say it's the PCT, proximal convoluted tubule. A lot of shed is reabsorbed there. So purple arrows signify secretion, so secretions from the blood to the filtrate, so back, so it goes into our urine for excretion. Orange arrows signify reabsorption. So hydrogen, NaCl, and nutrients are reabsorbed, but bicarbonate, water, ammonia, and K plus are secreted. So here we have an answer. Orange is, so purple is secretion. Purple is secretion. Orange is reabsorption. Let's see if we can turn this purple. Secretion first, reabsorption second. Secretion for bicarbonate, water, ammonium, and K plus. So we need, let's bring this here. We need mnemonics for secretion versus reabsorption. Solids. Then we have nephron function. The four processes of the kidneys are. We also need mnemonics for renin, angiotensin, aldosterone, and then ANP, anti-natriuretic peptide, for ADH, and there was also epinephrine and all the other ones, right? The four processes are filtration, which is from blood to lumen, reabsorption from lumen to blood, secretion from blood to lumen. So secretion is just anywhere else than glomerulus, filtration is at the glomerulus, and excretion from lumen to outside the body. This model nephron has been untwisted so that fluid flows left to right. Filtered 180 liters per day. What the hell? 100% volume, 300 osmolarity, and then we have reabsorption here. We have secretion here. We have reabsorption. So the startup loop of Henle is the same osmolarity, 30% volume, 54 liters per day. Then we have 18 liters per day, less liters per day for end of loop of Henle. 100. So the osmolarity is lower because we have so when the osmolarity is lower, it means that we have more water or less solutes in here, and that makes sense since we reabsorb a lot of shelling from the filtrate. So yeah, the osmolarity is lower, so a lot of stuff has been reabsorbed, and then we have a 50 to 1200 milliosmolarity for here, and 1.5 liters per day at the end of collecting duct to bladder and external environment. We have segment of nephron, renal corpuscle, glomerulus, plus Bowman's capsule. The processes is filtration of mostly protein-free plasma from the capillaries into the capsule. We have proximal tubule, so it's isoosmotic reabsorption of organic nutrients, ions, and water, secretion of metabolites and xenobiotic molecules such as penicillin. Then we have loop of Henle, reabsorption of ions in excess of water to create dilute fluid in the lumen. Countercurrent arrangement contributes to concentrated interstitial fluid in the renal medulla. Then we have distal tubule, regulated reabsorption of ions in water for salt and water balance and pH homeostasis. Then we have major segments of the nephron and their functions. The water permeability, extremely high for glomerulus, very high for PCT. For straight segments of PCT, very high. It has convoluted and straight segments. It's high and then it goes low, and then for the collecting duct and the cortical collecting duct, it's variable. Proximal. Okay. So the PCT is the sign we got this. What part of nephron is the site of secretion of H plus, urea, H3, and K plus? It should be secretion, so it's from the blood to filtrate. So what part of nephron is site of secretion for H plus, urea, NH3, and K plus? I mean, definitely PCT. Still, proximal convoluted tubule, a number of waste products are secreted from the blood into the PCT for excretion. H plus is 1, it's too acidic, you don't want blood to be too acidic. Urea, N and K, hunk, dump the hunk, H plus, urea, NH3, and K plus, dump the hunk. dump tank is amazing mnemonic. I love it. Dump the hunk. For PCT. What part of nephron is permeable to water but not salt? So first, water is so when you're going down, you want to get rid of the water, so you want to reabsorb it. Therefore, it is the descending limb of loop of Henle. But when you're going up, you're going back to the cortex of the kidney, so you want less. Less concentration. It's less concentrated urine, so you have reabsorption of salt, but not water. Therefore, as the filtrate moves into the more osmotically concentrated renal medulla, water is reabsorbed from the filtrate. It totally makes sense. What part of nephron and another part of nephron flow in opposite directions? It's descending and ascending loop of Henle, limb of loop of Henle, creating a countercurrent multiplier system that allows. What does the countercurrent multiplier system do? It allows for maximal reabsorption of water. Oh, so it's the vasa recta and loop of Henle. They flow in opposite directions. So we got this wrong. So the vasa recta blood network that surrounds the loop of Henle flows in the opposite direction, counter current of the loop of Henle. So vasa recta. Vasa is from veins, recta. What is the root of the word vasa recta? Countercurrent exchange. Cortex, low salt, high water. Medulla, high salt, low water. Countercurrent multiplication in the kidneys is the process of using energy to generate an osmotic gradient that enables you to reabsorb water from the tubular fluid and produce concentrated urine. This mechanism prevents you from producing liters and liters of diluted urine every day, and is the reason why you don't need to be continually drinking in order to stay hydrated. More salt is continually added by PCT. So the countercurrent system is why you don't need to drink so much water every day. The higher the osmotically of the more water leaves the descending limb of by osmosis. The more water that leaves the descending limb, the saltier the fluid is that remains in tubule. The saltier the fluid in the ascending limb, the more salt the tubule pumps into the ECF. The more salt that is pumped out of the ascending limb, the saltier the ECF is in the renal medulla. Long loop of Henle establishes a vertical osmotic gradient, countercurrent multiplier. The vasa recta preserve this. Huge note. The single effect of countercurrent multiplication process. So the single effect is when the filtrate is hypotonic with higher water potential. That happens in the ascending limb. The interstitium is now salty, so the medulla, like the outside of the filtrate, is now salty or hypotonic with a higher water, or hypertonic, and will attract water as below. Active transport of these ions from the thick ascending limb creates an osmotic pressure, drawing water from the descending limb into the hyperosmolar medullary space, making the filtrate hypertonic with a lower water potential. The countercurrent flow within the descending and ascending limb thus increases or multiplies the osmotic gradient between tubular fluid and interstitial space. So we need to know where is the interstitial space. So we have the tubular fluid. What is the tubular fluid? Then what is the single effect? Why is it called that? By what is the single effect? Why is it called that? By the counter current multiplier system. The kidney regulates pH by adjusting the levels of excreted H+ and reabsorbed bicarbonate. What part of nephron is permeable to salt but not H2O? It is the ascending limb to salt. It doesn't have aquaporins, as we read from the previous note, the big one. Therefore, salt is reabsorbed both passively and actively. So the passive reabsorption is here, but the active reabsorption, I believe, is in the PCT and DCT. But just to make sure. So this, I believe, is the concentration of the salt, as you can see. Where is salt transported? Actively and passively in the kidneys. Long loop of Henle establishes a vertical osmotic gradient. Countercurrent multiply. The vasorector preserve this gradient while providing blood to renal medulla. Countercurrent exchanger. Collecting ducts of all nephrons use the gradient in conjunction with the hormone vasopressin to produce urine of varying concentration. Osmotic equilibrating device. Collectively, this entire functional organization is known as medullary countercurrent system. So we have countercurrent multiplier, countercurrent exchanger, and osmotic equilibrating device, which is the vasopressin. The diluting segment of the nephron is in the outer medulla because what is actively reabsorbed in the side? It's salt. Oh, so actively reabsorbed is in PCT and DCT, as I imagine. Here it is, vasorecta, interstitial fluid osmolarity, interstitial is here outside of the vasorecta or the nephron. The interstitial osmolarity is different. You see that in the cortex is lower and the medulla is much higher. It's 300 in the cortex and 2700 in the medulla. In the inner medulla, in outer medulla, it's only twice, but here it's like I don't know, 9 times. What is it? 3 times 8 is 24. 3 times 9 is 27. Just as I said it. So we have active transport in the outer medulla, so in the ascending limb. Yeah. Amazing primary transporters and drug targets at apical membrane, non-sodium H, carbonic anhydrase, SGLT2, uric acid, and base transporters, aquaporins. So aquaporins is mainly the feature for descending limb, but for ascending limb, it's sodium, potassium, NKCC2, NCC, NAC, then the medullary collecting are aquaporins, vasopressin, diuretic with major action, vasopressin antagonists. Diuretic means you have more water in your urine, so when you're antagonizing vasopressin, ADH, it means that water is not reabsorbed, so you have it more in your urine. Makes sense. Aldosterone is a hormone synthesized in the adrenal cortex in response to angiotensin 2 or high K plus concentration. What part of nephron is responsive to aldosterone and is the site of salt reabsorption and waste product secretion? So I believe, so it is both the DCT and collecting duct that are responsive to aldosterone, and they reabsorb salt, and so distal, so it's only DCT that is a site of salt reabsorption and waste product secretion, similar to PCT. What part of nephron has variable permeability, which allows the reabsorption of the right amount of water depending on the body's needs? Variable permeability, I would say, so it's definitely not the descending limb. It should be the collecting duct. Yeah, it's responsive to both aldosterone and ADH. It allows reabsorption of the right amount of water depending on the body's needs. It's the collecting duct. So collecting duct, duct tape, water reabsorption, vase, sponge pores, aquaporins. give the 3 skin layers. The outer is epidermis, the middle is dermis, and the inner is hypodermis, subcutaneous layer. Amazing. We have fatty tissue under the hypodermis. With hypodermis. We have blood vessels. Over it we have hair follicle. Until the dermis we have oil gland. It's here. We have lymph vessels. We have sweat glands, and yep. Most of the skin can be classified as thick skin, thin skin. Yes, it's thin skin. My bad. Thick skin has an extra layer, lucidum, but that is not the reason that it is thicker than thin skin. Real reason is the other layers are thicker in thick skin than in thin skin. But most of the skin can be classified as thin skin. So thin skin we have here, epidermis, dermis. Thick skin we have here is like no epidermis. This is epidermis apparently, and this is dermis. We have Langerhans cells here. We have laminar granules. We have dead keratinocytes. We have the stratum corneum, stratum lucidum. We have granulosum. We have spinosum. We have stratum germinativum. I didn't see this. Basement membrane. Then we have dermis. So between the dermis and hypodermis, epidermis, there is the basement membrane. We have keratinocytes. We have melanocytes. We have tactile disc. We have Merkel cell. Yeah, we need to know what these are. So we have the basement membrane. Like here, we have the P. We have the R. We have the D. Okay. Cool. It's 9:55. I really want to finish this. But I feel like there's light at the end of the tunnel. What is the muscular lining of bladder? It relaxes to store urine and contracts during urination. It's the detrusor muscle. Thick skin is found only on what? And on the palms of the hands and the soles of the feet. Thick skin is only on the palms of the hands and the soles of the feet. Thin skin epidermis has the following 4 layers. For the thick skin epidermis, you would add stratum lucidum right between stratum corneum and granulosum. So we have the basale, which is the inner epidermis, and then for the outer one, we have stratum corneum, granulosum. Between cornea and granulosum, you can, for thick skin, add the lucidum and then spinosum. So it's C. So we have CLGSB. CLGSB. CL, no L, G, S, B. CL, corneum, lucidum, granulosum, S, spinosum, basale. What is the epidermis layer that is only found in thick skin? Is lucidum. Stratum lucidum. What are so lucidum thick, lucid thick, thick lucids, thick lucids, lucid, thick LSDs. lucid. What are a type of dendritic cells found in the skin? They present antigen to T cells. It's defensins. Langerhans cells. My bad. They present antigens to T cells. Langerhans cells. Under skin. First line of defense are Langerhans cells, epidermis, LCs, subtype of dendritic cells. Engulf the antigens in order to present it to T cell. We have Langerin, CD207, the most important surface molecule. C-type lectin resembles DC sign CD209 expressed by DCs. Dendritic cells. What is produced by melanocytes and protects skin from DNA damage caused by UV radiation? It is melanin. This is produced by melanocytes and protects skin from DNA damage. Melanin, UV rays, skin layer, dark spots. We have skin cell exposed to UV light. We have melanocytes, then melanin, and then dark spot. Definitely want to watch the YouTube video for this. Give the two layers of the dermis. We have papillary and reticular. Papillary, reticular. Papillary, reticular. Papillary, reticular. Papillary, reticular. Hypodermis, subcutaneous tissue, not part of the skin. It's interesting. It's not part of the skin. Appendages of skin: eccrine sweat gland, erector pili muscle, sebaceous gland, oil gland, hair follicle, hair roots. What are mechanoreceptors essential for light touch sensation? Mesenchymal cells. Ooh, I got it right. But we gotta do it again. It's no use. Yeah, I want to do skin later, to be honest. So it's 10 p.m. Let's take a rest and be back.